everyone. Today I am here, Dr. Megha Pan will teach you about the protein biochemistry. Proteins. So what are proteins? Proteins are the biomolecules that are present in every part of a body. They contain carbon, oxygen, nitrogen, hydrogen and sulfur. These proteins are made up of large amino acids. These amino acids are joined together by the carboxyl group of one amino acid and amino group of the next through peptide bond. Through peptide bond, these amino acids are joined together to form the linear chains of the proteins. Some proteins are comprised of only one polypeptide chain, while others can be of two or more polypeptide chains. Talking about the size and shape of proteins, the number, type and arrangement of amino acid differ in different proteins. So, here as we can see, the proteins with different molecular weight, number of residues and the difference in the number of polypeptide chains. Here the chymotrypsin has the number of polypeptide chains present is 3, whereas in hemoglobin it is 4. Talking about the general properties of proteins, these proteins are high molecular weight substances. They can be of different shape. These are globular proteins and the fibrous proteins. Globular protein, as we know, are soluble in water, whereas the fibrous protein are insoluble in water. Proteins are amphoteric compounds. They are present as the acid and the base in aqueous solution, and at isoelectric point they precipitate. What is the isoelectric point? At the neutral pH these proteins precipitate. Now we will talk about the classification of proteins. Proteins can be classified into 3 classes on the basis of their chemical composition. Second is the shape and third one is on their biological function. Proteins according to the chemical composition can be classified into 2 main classes. First is the simple proteins and second is the conjugated proteins. So, what are simple proteins? Simple proteins are proteins which upon hydrolysis give only amino acids. Example are chymotrypsin, ribonuclease. Whereas the conjugated protein upon hydrolysis will give amino acid and the prosthetic group. The prosthetic group is the non-protein part present in the conjugated proteins. Conjugated proteins can further be classified on the basis of their prosthetic group attached. If it is the carbohydrate part, it is known as glycoproteins. Lipid part, it is known as lipoproteins. Heme contains called hemoproteins. Metal containing known as metalloproteins. Now, here we will study about some examples of conjugated proteins. So, lipoproteins. Here the prosthetic group is the lipid. Example is beta-1 lipoprotein of blood. Glycoproteins, the prosthetic group we know, it's carbohydrate. Example is immunoglobulins. IgG is one of the example. Flavoproteins, its prosthetic group is flavin nucleotides. Example is the succinate dehydrogenase enzyme. Whereas in metalloproteins, Example is the alcohol dehydrogenase enzyme. Now, according to their shape, proteins can be classified on the basis of globular proteins and the fibrous protein. So, globular proteins are, these are proteins soluble in water and folded into a globular shaped structure. Examples of globular proteins are hemoglobin and myoglobin. Fibrous proteins, tough proteins, these are insoluble in water. Example is the structural proteins, that is the collagen, elastin and keratin. Now, we will classify these proteins on the basis of their biological function. On the basis of their functions, these can have a catalytic function, they can be enzymes, can be transport function. Transport proteins are the hemoglobin, albumin and transferrin. Contractile proteins, these are the actin and myosin. Structural proteins which provide the structure are the keratin, elastin, collagen. Then we have the defense proteins, IgG, immunoglobulins, fibrinogen, regulatory functions. Some hormones are proteins like the growth hormone. Somatotropin is a protein hormone. Defense in antibodies and coagulating factors can be proteins. Now, talking about the protein structure. The amount and type of amino acids determine the structure of a protein. The amino acid sequence of a number of proteins has been determined, and it is established that the sequence of amino acid is the force that determines the protein conformation. So, on the level of their classification, protein structure can be considered at 4 levels. First is the primary structure, then the secondary structures, tertiary structure and the quaternary structures. This is the primary structure of the protein, a linear polypeptide chain. Then we have the secondary structure, can be alpha-helix, a spiral helix and a beta-pleated elongated sheets. Tertiary structure, folded protein into a 3D conformation is known as the tertiary structure. Then more than 2 polypeptides joined together known as the quaternary structure. The forces that stabilize the different protein structure. If we talk about the primary structure, the major interaction is the covalent bond formed between the amide and the carboxyl group, the peptide bond. Then is the secondary structure that stabilize, which bond stabilize the secondary structure? It's the hydrogen bonds. Then the 3D structure, tertiary structure, ionic bonds, disulfide bonds, hydrophobic interaction and hydrogen bonding stabilize the tertiary structure. And the quaternary structure is also stabilized by the disulfide bond, hydrophobic interaction and hydrogen bonding. Now, if we talk about the primary structure, it is the sequence of, linear sequence of amino acid present in a protein, and different protein differ from each other in their primary structure. The bonds responsible, as we have already seen, what are these bonds? For the primary structure are the covalent bond. These can be the peptide bonds formed between the carboxyl group of one amino acid and the amino group of another amino acid, and the disulfide linkages or bonds formed between two cysteine residues. Then the secondary structure. Secondary structure, the linear sequence is arranged into the alpha helix, beta-pleated sheet and the collagen helix. Then the tertiary structure, we have already seen that the tertiary structure is the three-dimensional conformation. The protein is folded in the 3D structures. Here we can see, as we can see here, the hydrophobic interaction in the interior of the protein and the disulfide bridges stabilize the 3D structure, tertiary structure of the proteins. Then the quaternary structure. Example of the quaternary structure is more than two polypeptide chain linked together forms the quaternary structure. Quaternary structure example, as we can see in the molecule of hemoglobin. Bonds that stabilize quaternary structure, we have already seen the hydrophobic interaction, hydrogen bonds and the ionic interaction. Now we will study about some protein deficiency diseases. These are the diseases which can be due to the deficiency of the proteins. These are the causes for, it is a protein deficiency which affect the growth and development. The symptoms include swelling in the legs and abdomen and skin pigmentation. Marasmus is also a deficiency of protein, can be seen in malnutrition people. Here the symptoms include weakness, muscle fatigue and a loss of muscle mass. Edema, what is edema? Edema is a condition in which the body is filled with fluid. If the body is filled with fluid, what will happen? We can see swelling in the ankles, legs and feet. Then protein deficiency can impair our immune system. Our immune system, our ability to fight off infections can be severely infected by the deficiency of proteins. And it can also be seen that those children which have the deficiency of protein, decreased growth can be seen, decreased growth and development can be seen in this kind of children. Thank you.